the basic fundamental idea here is that all cells that we're familiar with, that's animal cells, plant cells, fungi, mushroom, yeast, those kinds of cells, protist cells, that means like algae and slime mold cells, all of these large cells with their nuclei, nucleated cells that have the chromosomes that we count and so on, those kinds of cells are all like mixotricha. They are all composites. They, in some level, are individuals, but their individuality is the result of fusion of evolutionary branches on evolutionary trees, not the result of divergence and bifurcation, which is the usual topology, the usual model for evolution. So that's the basic idea. But the serial endosymbiotic theory is can be detailed, and the basic way to start the details is that the components are all once individual bacteria, but the bacteria joined and fused in a specific order. That's what the serial part comes from. And endo comes from the ideas that bacteria are actually inside of bacteria. This has become very interesting recently because in the last month, some people from Utah have published a paper showing that inside of insects, these bodies that they thought were organs are really bacteria inside of another bacterium, the normal insect. And so it means that this kind of process of bacteria getting inside of bacteria is not limited to our ancestors' nucleated cells, but it happened over again in the evolution of important insects. At any rate, so the steps go as follows, and we've had a big breakthrough in it, I think. The first steps were under completely anoxic conditions, which means no oxygen in the atmosphere. They were just in these organisms that are poisoned by oxygen. So you have archaebacteria, which is one kind of bacterium, and eubacteria, a swimming eubacterium, and probably a sulfide, hydrogen sulfide producing archaebacteria. And these organisms formed a consortium. That means they live together. And the swimmer moved the other one by just moving around all the time. What is exciting there is that an exact consortium like that has been discovered by a Russian investigator called Dubinina. And in fact, it was in the literature from 1993 on, but we didn't, it was in the Russian literature, and we didn't get it translated until much more recently. And we realized that what we are hypothesizing, by we, I mean here, Dennis Searcy, a colleague of mine at the University of Massachusetts, and myself, what we have been hypothesizing has actually been seen in nature, seen in quantity. It was called phyo-dendron-laytons, as if it were one bacterium, and with good analysis it turned out to be two bacteria very different from each other, and one of them a spirochete, which is what I hypothesized was the origin of cilia. It's really very exciting. Now, I'm fond of saying that we've won 3 out of 4. We've won the mitochondria. There's no doubt the mitochondria actually are alpha-perennial bacteria. Everybody agrees who's looked at the problem. We've won the plastids. That was the easiest to win because the plastids are green and you can see them and they have free-living analogs. And when plastids were first described in the 19th century, people who described them said they're very much like they probably came from symbionts. They're very much like the free-living cyanobacteria that are right next door. So we won the cyanobacteria, we won the plastids. We've won the archaebacterial origin of the rest of the cell, and the piece left is the motile structure. Did cilia come from spirochetes? And since we have 2 spirochetes, infamous of course, the one that causes Lyme disease, called Borrelia, and the one that causes syphilis, called Treponema pallidum. Those 2 spirochetes have recently been sequenced. Full sequence, that means we have all the DNA from both of those. We can start looking to see if in that DNA there's the DNA that codes for the proteins that we see that would have homology, that would be related to the motile proteins involved in all other cells. In other words, the new molecular biology is giving us the tools to definitively win 4 out of 4 or not. And I think it's just a question of time and doing the work.